Hello everyone, I am Dr. Trupti and you are watching my YouTube channel Enjoy Biochemistry. In the video lecture series on nucleotide metabolism, today let's learn about biosynthesis of purine nucleotides. At the end of this session, students shall be able to describe de novo biosynthesis of purine nucleotides in detail with its regulation. Let's first see the difference between nucleoside and nucleotides. When the nitrogenous base is linked to the sugar molecule, it becomes nucleoside. And when this nucleoside is linked to the phosphate, it becomes nucleotide. So nucleotide has nitrogenous base, sugar and phosphate. And the pentose sugar is D-ribose in ribonucleotides of RNA, while in deoxyribonucleotides of DNA, the sugar is deoxyribose. These nucleotides, they participate in almost all the biochemical processes either directly or indirectly. They are the structural components of nucleic acids like DNA, RNA, and also the coenzymes, and they are involved in regulation of several metabolic reactions. Now the nitrogenous base present in the nucleoside and nucleotides, they can be purines like adenine and guanine or pyrimidine like cytosine and thymine. So if we take example of adenine, adenine is a purine. When this adenine is linked to the sugar, that is pentose sugar, it becomes nucleoside, and when it is further linked to the phosphate group, it becomes adenosine monophosphate. It is also called as adenylate acid, and that adenylate acid or AMP, it is the nucleotide. This is the structure of purine and it contains two rings and it is numbered as 1, 2, 3, 4, 5, 6, 7, 8 and 9. At position 1, 3, 7 and 9, nitrogen is present. At position 2, 4, 5, 6 and 8, carbon is present. The two purines commonly found in nucleic acid are adenine and guanine, and both are found in DNA as well as RNA. Other purine metabolites which are not usually found in nucleic acids, they include xanthine, hypoxanthine and uric acid. So this is the structure of purine adenine. It is also called as 6-aminopurine because at the 6th position amino group is linked. And this is the structure of guanine. It is called as 2-amino-6-oxypurine because at the 2nd position amino group is attached and at the 6th position oxygen is attached. So guanine is called as 2-amino-6-oxypurine. So there are two purines, adenine and guanine. This is the structure of nucleotide, that is adenosine monophosphate. Here you can see this is the adenine which is linked to this ribose sugar by beta and glycosidic bond which is formed between N9 of adenine to C1 prime of this ribose sugar. This is the ribose sugar, and the carbon atom of this ribose sugar, they are denoted by using a prime number like 1 prime, 2 prime, 3 prime to avoid the confusion with the carbon atoms of purine ring. So this is the adenosine monophosphate, or it is also called as adenylic acid. If adenine is the nitrogenous base, then purine nucleotide is adenosine monophosphate, and 2'-deoxyadenosine monophosphate is the deoxyribonucleotide. If guanine is the nitrogenous base, then ribonucleotide is guanosine monophosphate and deoxyribonucleotide is 2'-deoxyguanosine monophosphate. Let's see the overview of purine biosynthesis. Humans can synthesize both purines and pyrimidines, and that's why the dietary intake of nucleic acid and nucleotides is non-essential. The purine biosynthesis can occur in three ways. The first and most important is de novo synthesis of purines, second is phosphoribosylation of purines, and third is phosphorylation of purine nucleosides. Out of these three, the most important is de novo synthesis of purines, which occurs in the liver. And this ribose 5-phosphate is the starting material for synthesis of purines. And these purines are not synthesized individually like the synthesis of adenine and synthesis of guanine. They are synthesized, or these purines are built upon a pre-existing ribose 5-phosphate. So the ribose 5-phosphate is the starting material and it is derived from the pathway, that is the HMP pathway, hexose monophosphate pathway. And there are about 11 steps through which the first purine nucleotide that is formed is inosine monophosphate, that is the IMP. And that's why it is called as parent nucleotide, because it is first synthesized and later on the other nucleotides like AMP and GMP are synthesized. So from this IMP further there is formation of AMP, that is adenosine monophosphate, or GMP, that is the guanosine monophosphate occur. And from this AMP further there is formation of adenosine diphosphate, that is ADP, and adenosine triphosphate, that is ATP occur. And from GMP, GDP and GTP are formed. Now the deoxyribonucleotides, they are formed from the ribonucleotides like adenosine diphosphate and guanosine diphosphate will result in the formation of deoxyadenosine diphosphate and deoxyguanosine diphosphate. So this is the overview of purine biosynthesis which occurs in liver. What are the various sources of carbon and nitrogen present in the purine ring? This is very important for MCQ as well as BAQ, that is brief answer question. So in this purine ring, the N1 of purine, it is obtained from amino group of aspartate. C2 and C8, they are obtained from formate of tetrahydrofolic acid. Then N3 and N9, they are obtained from amide group of glutamine. C4, C5 and N7, they are contributed by glycine, and C6 directly comes from carbon dioxide. Before going into details of all the steps of purine biosynthesis, let's first see the various products that are formed in the purine biosynthesis, and everything about this purine biosynthesis steps is given in this one table. So if you remember this table, if you understand the names of products which are formed in the steps, then you can easily write the purine biosynthesis pathway. So you know that for the purine biosynthesis, various amino acids are required like glutamine, glycine, aspartic acid, then folic acid is also required, carbon dioxide and ATPs are required. So the first starting material that is required for this purine biosynthesis is ribose 5-phosphate, because purines are built on the pre-existing ribose 5-phosphate, and this ribose 5-phosphate, it is derived from the HMP pathway. So there are 11 steps. In the first step, ATP is involved, so ATP is incorporated and this ATP is converted to AMP. So 2 phosphate groups are donated to this ribose 5-phosphate and there is formation of phosphoribosyl pyrophosphate. So this is the first product that is formed, which is also called as PRPP, that is the phosphoribosyl pyrophosphate, which is formed in the first step of purine biosynthesis. Now in the second step, glutamine is involved and this glutamine is the donor of N9. So it donates this nitrogen is donated by the amide group of this glutamine. So it becomes phosphoribosylamine because it is the glutamine amino acid, that's why it becomes phosphoribosylamine. In the third step, glycine and ATPs are required and this glycine is responsible for this contribution of this C4, C5 and N7. So when the glycine is there, the product will become glycinamide ribosyl 5-phosphate. So now why the name is glycinamide ribosyl 5-phosphate? Because here the glycine in the third step, glycine is the donor which donates this C4, C5 and N7, and that's why it is the glycinamide ribosyl 5-phosphate. Now in the fourth step, methynyl tetrahydrofolic acid is involved and this is responsible for this formation of or contribution of this carbon which is present at number 8, and this is the donor, one carbon donor, that is the donor of formyl group. So this product name will include this formyl word. So in the fourth step, the product formed is formyl glycine. The same thing from the third, only formyl word is attached. Formyl glycinamide ribosyl 5-phosphate. Now coming to the fifth step. In the fifth step, glutamine is the donor ATP is required, and this is responsible for the nitrogen which is present at the third number. So again it becomes the formyl glycinamidine. Here it is glycinamide. Now only changes, it becomes glycinamidine ribosyl 5-phosphate. The other name is same, only because it donates the nitrogen, it becomes formyl glycinamidine ribosyl 5-phosphate. In the sixth step, ATP is required. There is closure of the ring and there is formation of imidazole ring, and that's why the product is amino-imidazole ribosyl 5-phosphate, because the imidazole ring is formed with the help of ATP and there is ring closure. The first ring is formed, so it is the amino-imidazole ribosyl 5-phosphate. In the seventh step, carbon dioxide is incorporated, which is responsible for the contribution of carbon present at the sixth number. So now the name of product is amino-imidazole, same from the previous name, only as the carbon dioxide is donor, and here it becomes carboxylate 5-phosphate. So carboxylate because carbon dioxide is involved here. Now in the eighth step, aspartic acid is involved, which contributes this N1. ATP is also required here, and here the product form is amino-imidazole succinyl carboxamide ribosyl 5-phosphate, because aspartic acid, and that's why it is the succinyl, because succinyl group is added there. In the ninth step, nothing is added, only fumarate is removed from this product, and so succinyl is removed. Now it becomes only amino-imidazole carboxamide ribosyl 5-phosphate. In the 10th step, formyl tetrahydrofolic acid is the donor, and it is responsible for the contribution of carbon present at the number 2. So formyl group is donated. So again the name will have some form amino-imidazole carboxamide ribosyl 5-phosphate. So here at the 10th step, the product form is form amino-imidazole. Only form is added here because formyl group is donated. Form amino-imidazole carboxamide ribosyl 5-phosphate. In the 11th step, there is closure of the second ring and the product form is Adenosine Monophosphate, IMP. So there are 11 steps. If you remember this chart, if you remember the donors and added carbon number, and if you correlate the names with the donor, then you can easily remember. Though the names of product looks scary, but with practice and with revising, you can remember all the names of products of purine biosynthesis. Now let's see each step of purine biosynthesis in detail. So the first step was synthesis of phosphoribosyl pyrophosphate, that is the PRPP, and from the starting material was ribose 5-phosphate, and this ribose 5-phosphate, it is the product of HMP pathway. We get this ribose 5-phosphate from HMP pathway, and it is the starting material for biosynthesis of purine nucleotides, because purines are built upon the pre-existing ribose 5-phosphate. So in the first step, what is involved? ATP. So here ATP is converted to AMP by the action of enzyme PRPP synthetase. Whenever there is involvement of ATP, the enzyme will be synthetase. Remember that. So the enzyme is PRPP synthetase because the product form is PRPP, that is phosphoribosyl pyrophosphate, and in this step it is synthesized. That's why the name of enzyme is PRPP synthetase. And this is the structure of PRPP, that is the phosphoribosyl pyrophosphate, where you can see that 2 phosphate groups are linked here in the PRPP structure, and that's why from ATP 2 phosphates are donated and there is formation of AMP. So this is the first step, that is synthesis of phosphoribosyl pyrophosphate. And AMP and GMP, these are the inhibitors of this PRPP synthetase, and inorganic phosphate, it is the activator of this PRPP synthetase. The second step of purine biosynthesis is synthesis of phosphoribosylamine. Here the glutamine is the nitrogen donor, and this is responsible for contribution of nitrogen present at the 9 number. So this phosphoribosylamide, it is synthesized from the product which is formed in the first step, that is PRPP. So on this PRPP, there is addition of glutamine, there is formation of glutamate and release of pyrophosphate. This reaction is catalyzed by glutamine PRPP amido transferase. So there is transfer of amido group. That's why it is the amido transferase. Glutamine transfers, that's why it is the glutamine to PRPP. That's why glutamine PRPP amido transferase. So this pyrophosphate is removed and there is addition of this amino group and it becomes phosphoribosylamine. So this is the second step of purine biosynthesis. And this availability of substrate, here the substrate is PRPP. So if there is PRPP is increased or there is availability of PRPP, it will activate this enzyme glutamine PRPP amido transferase and there is formation of phosphoribosylamine. And this enzyme is inhibited by feedback regulation or feedback inhibition by its product like IMP, AMP and GMP. By feedback inhibition, they can regulate or they can inhibit this enzyme glutamine PRPP amido transferase, and this is the committed step of purine biosynthesis. So this is the second step, that is synthesis of phosphoribosylamine. Here the glutamine is donor which donates this N9 of this purine ring. Third step of purine biosynthesis is synthesis of glycinamide ribosyl 5-phosphate. Here glycine is the donor of C4, C5 and N7. So from phosphoribosylamine, the glycine is added with the help of ATP and that's why the enzyme is synthetase. There is formation of ADP and inorganic phosphate and it leads to formation of the product in the step 3, that is called as glycinamide ribosyl 5-phosphate. So in the third step, the glycine contributes carbon number 4, carbon number 5 and nitrogen present at number 7. Step number 4 is synthesis of formyl glycinamide ribosyl 5-phosphate. And formyl, so tetrahydrofolic acid is donor of C8. So this carbon 8 is donated in the fourth step. So glycinamide ribosyl 5-phosphate, there is transfer of formyl group from this N5-N10 methynyl tetrahydrofolate and there is transfer of formyl group, that's why the enzyme is formyl transferase, and there is formation of formyl glycinamide ribosyl 5-phosphate. Only formyl word is added here and here the addition of formate group, and that's why the product is formyl glycinamide ribosyl 5-phosphate. So in this fourth step, it is responsible for contribution of this carbon which is present at the 8th position. Step 5 is synthesis of formyl glycinamidine ribosyl 5-phosphate. So the contributor is glutamine which is responsible for contribution of nitrogen present at the third number, number 3. So here from formyl glycinamide ribosyl 5-phosphate, glutamine is added. It also requires ATP, so the enzyme is synthetase. There is formation or release of glutamate, ADP and inorganic phosphate, and amide group is added here. So it becomes formyl glycinamidine ribosyl 5-phosphate. The glycinamide becomes glycinamidine and here the nitrogen is added in the purine ring structure. So the step 5 is formation or synthesis of formyl glycinamidine ribosyl 5-phosphate which is responsible for contribution of N which is present at the third position. Step number 6 is synthesis of amino imidazole ribosyl 5-phosphate. So in this reaction there is the ring closure. So this formyl glycinamide ribosyl 5-phosphate. So now this is the ring of purine. So the ring closure will occur in this reaction. ATP is required here. That's why the enzyme is synthetase. And there is ring closure and closure of ring will lead to formation of imidazole ring. And that's why the product is amino imidazole ribosyl 5-phosphate. So this is the step number 6 where the imidazole ring, this is the structure of imidazole ring and the purine ring has one imidazole ring and that's why the product form is amino imidazole ribosyl 5-phosphate. So step 6 of purine biosynthesis, there is formation of this imidazole ring. Coming to the step number 7, there is synthesis of amino imidazole carboxylate ribosyl 5-phosphate. So carbon dioxide is donor for this carbon present at the 6th number. So this amino imidazole ribosyl 5-phosphate, there is addition of carbon dioxide by the enzyme carboxylase. This is the carboxylation reaction, and there is formation of 5 amino imidazole. Only the word carboxylate is added here, carboxylate ribosyl 5-phosphate. So this is the carboxylate group which is added here, and you can see the carbon dioxide contributes this carbon number 6, and there is formation of amino imidazole carboxylate ribosyl 5-phosphate. And in this carboxylation reaction, when carbon dioxide is added, no ATP is required, no biotin is required. However, in other carboxylation reaction, you see, for example, in the pyruvate carboxylase, when there is formation of oxaloacetate from pyruvate, there is requirement of ATP, biotin. But in this carboxylation reaction, there is no requirement of ATP or biotin. So in the step number 7, there is formation of amino imidazole carboxylate ribosyl 5-phosphate, which is responsible for the contribution of carbon present at number 6. Step number 8 is synthesis of amino imidazole succinyl carboxamide ribosyl 5-phosphate. So succinyl is here. So aspartate is the donor responsible for contribution of nitrogen at one position. So from the amino imidazole carboxylate ribosyl 5-phosphate, here aspartate and ATP is involved. So the enzyme is synthetase, and there is formation of amino imidazole succinyl carboxamide ribosyl 5-phosphate. Only succinyl group, and it becomes carboxamide. So this is the succinyl group of aspartate, and this is step number 8, which is responsible for contribution of N present at number 1. Step number 9 is synthesis of amino imidazole carboxamide ribosyl 5-phosphate. So succinyl word is gone from here, and in this 9th step, there is no addition, only fumarate is removed, and by the action of enzyme adenosuccinate lyase, and now it becomes amino imidazole carboxamide ribosyl 5-phosphate. So succinyl, the word is not here. There is release of fumarate in the step number 9. So step number 9 is formation of amino imidazole carboxamide ribosyl 5-phosphate. Step number 10 is synthesis of form amino imidazole carboxamide ribosyl 5-phosphate. So the formate group is transferred here by tetrahydrofolic acid, and it is responsible for contribution of carbon number 2. So from this amino imidazole carboxamide ribosyl 5-phosphate, there is transfer of formate group from N10 formyl tetrahydrofolate by the enzyme formyl transferase, because there is transfer of formyl group, that's why the enzyme is formyl transferase. And you can see here there is formation of amino imidazole carboxamide ribosyl 5-phosphate. So this carbon number 2, you can see this carbon number 2, it is contributed by this formyl tetrahydrofolate in the step number 10. So in the step number 10, the formyl tetrahydrofolate donates or contributes the carbon which is present at position 2. Coming to the last step of purine biosynthesis, that is step number 11, there is synthesis of parent purine nucleotide, that is adenosine monophosphate or IMP. So from this form amino imidazole carboxamide ribosyl 5-phosphate, there is just release of water molecule by action of enzyme cyclohydrolase. There is ring closure, and there is formation of adenosine monophosphate. So this is the last step of purine biosynthesis, which involves the formation of parent purine nucleotide adenosine monophosphate. Now let's see how AMP is synthesized from IMP. IMP is the parent nucleotide, and from this IMP, AMP is synthesized. So in the first reaction, aspartate and GTP is involved. The enzyme is adenyl succinate synthetase. Adenyl succinate is formed, that's why adenyl succinate synthetase is the enzyme. So from AMP, adenyl succinate is formed, and later from this adenyl succinate, there is release of fumarate by enzyme adenyl succinase, which will cleave this fumarate from this adenyl succinate, and there is formation of AMP, that is the adenosine monophosphate. That's how AMP is from IMP, and the intermediate is adenyl succinate. Now from this AMP, ADP can be formed by AMP kinase. Here ATP is the phosphate donor, and from this ADP, ATP is formed by ADP kinase, and here the ATP is phosphate donor, which means that for the synthesis of ATP, ATPs are required. Synthesis of GMP also occurs from IMP. From this IMP, by action of enzyme IMP dehydrogenase, there is formation of xanthosine monophosphate, which is also called as XMP. From this XMP, there is formation of guanosine monophosphate. In this reaction, glutamine is added, and ATP and water molecule is involved in this reaction, and the enzyme is guanosine monophosphate synthetase, and there is formation of guanosine monophosphate from xanthosine monophosphate, which is synthesized from adenosine monophosphate. And from this GMP further, there is formation of GDP by action of enzyme GMP kinase by phosphorylation, and then GDP is phosphorylated to form GTP by GDP kinase. So this is how from IMP first GMP is synthesized, and from GMP GDP, and from GDP GTP are formed. So far we have seen the formation of ribonucleotides. Now let's see how these deoxyribonucleotides are formed. So the formation of deoxyribonucleotides diphosphate occurs from the ribonucleoside diphosphates like ADP and GDP. From this ADP and GDP, by the action of enzyme ribonucleotide reductase, there is formation of 2'-deoxyribonucleoside diphosphate. And in this reaction, reduced thioredoxin is involved, which is oxidized. And to maintain the thioredoxin in the reduced form, the NADPH is important, and this NADPH, by the action of enzyme thioredoxin reductase, it is responsible for maintaining this thioredoxin in the reduced form. And this enzyme complex, ribonucleotide reductase, thioredoxin reductase, this is functional only when cells are actively synthesizing the DNA, that is in the S phase of cell cycle. So whenever there is DNA synthesis, the requirement of deoxyribonucleotide is there, and in that condition only there is formation of deoxyribonucleotides occur. Purine biosynthesis regulation involves two mechanisms, that is the availability of substrate and feedback regulation. So substrate for purine biosynthesis are ribose 5-phosphate, and the first product that is formed, that is phosphoribosyl pyrophosphate. So availability of ribose 5-phosphate and phosphoribosyl pyrophosphate, this substrate will decide the rate of purine biosynthesis. So if there is more availability of PRPP and ribose 5-phosphate, there is more formation of purine biosynthesis. The second mechanism is feedback regulation. So this feedback regulation involves IMP synthesis, that is parent nucleotide synthesis, and from this IMP further AMP and GMP is synthesized. So at the IMP synthesis level, two enzymes are important. First is the PRPP synthetase, which leads to formation of PRPP, and second is glutamine PRPP amidotransferase, which is responsible for synthesis of phosphoribosylamine. And the products like AMP, GMP, they are the inhibitors of these enzymes. So IMP synthesis is regulated by feedback inhibition by AMP as well as GMP. Then the regulation also occurs at the AMP, GMP synthesis level, and adenyl succinate synthetase is the enzyme involved in the AMP synthesis. So AMP is the feedback inhibitor of this adenyl succinate synthetase, and IMP dehydrogenase is the enzyme of GMP synthesis. So GMP is the inhibitor of this IMP dehydrogenase. For purine biosynthesis, ribose 5-phosphate is the starting material, and step 1, which involves the enzyme PRPP synthetase, which leads to formation of PRPP, and second step is the committed step, which is catalyzed by glutamine PRPP amidotransferase, which leads to formation of phosphoribosylamine. These two steps are very important for purine biosynthesis, and further through series of reaction there is formation of AMP and then formation of first IMP, and then from the IMP, AMP and GMP is formed, and from AMP further ADP and ATP are synthesized. From the GMP, GDP and GTP are synthesized. So this AMP and ADP, they are the inhibitor, feedback inhibitor of this PRPP synthetase. GMP is the inhibitor of both PRPP and glutamine PRPP amidotransferase, while this GDP is the inhibitor of PRPP synthetase. From the IMP, first there is formation of adenyl succinate by adenyl succinate synthetase, and from the this IMP, xanthosine monophosphate is formed by IMP dehydrogenase, and from this xanthosine monophosphate, GMP is formed from the GMP synthetase. And further there is formation of AMP and GMP, and in this reaction AMP is the inhibitor of this adenyl succinate synthetase, and that's how it regulates its own synthesis by feedback mechanism. GMP is inhibitor of IMP dehydrogenase. So if there is ample amount of GMP, it will inhibit this enzyme IMP dehydrogenase. ATP is the activator of GMP synthetase, and GTP is the activator of this adenyl succinate synthetase. So this is how purine biosynthesis is regulated. Now let's see the regulation of deoxyribonucleotide synthesis. So the synthesis of deoxyribonucleotide occurs from the ribonucleotide, diphosphate, ADP and GDP. There is formation of deoxy-ADP and deoxy-GDP by the action of enzyme ribonucleotide reductase, and this enzyme is multi-subunit protein having 2 R1 and 2 R2 subunit. Now this R1 subunit has activity sites, which are the allosteric sites, other sites from the catalytic site, and it also has substrate specificity site, and when the ATP binds to this activity sites, it activates the enzyme and there is formation of deoxyribonucleotides. So ATP is the activator, and when deoxy-ATP means it binds to this activity site, there is inhibition of this ribonucleotide reductase enzyme. And various other activators like ATP, DATP, DTTP and DGTP, they regulate the reduction of specific ribonucleotides. So this is how the deoxyribonucleotide synthesis is regulated. The drug hydroxyurea, that is hydroxycarbamide, is the inhibitor of this ribonucleotide reductase enzyme, and that's how it inhibits the generation of substrates which are required for DNA synthesis, and this drug is used as the anti-neoplastic agent. Let's summarize today's topic, that is de novo synthesis of purine nucleotides, and this purine biosynthesis has 11 steps. There is first formation of IMP, that is adenosine monophosphate, and from this IMP, AMP and GMP can be formed. This purine biosynthesis occurs in the liver. The starting material is ribose 5-phosphate, which is derived from the HMP pathway, and purine ring is built upon the pre-existing ribose 5-phosphate. So there are 11 important steps. So the first step, there is formation of phosphoribosylamine pyrophosphate. ATP is the phosphate donor here. So in the first step, there is formation of phosphoribosylamine pyrophosphate. The second step, glutamine is the nitrogen donor, that is amide group is donated by the glutamine, which is responsible for the contribution of nitrogen present at number 9, and there is formation of phosphoribosylamine. So in the second step, there is formation of phosphoribosylamine. Third step involves glycine and ATP. The glycine is responsible for contribution of C4, 5 and nitrogen 7. So there is formation of glycinamide ribosyl 5-phosphate. This is the glycinamide ribosyl 5-phosphate. Fourth step, there is formation of formyl glycinamide ribosyl 5-phosphate. So here the formyl group donor is methynyl tetrahydrofolate. It is responsible for contribution of carbon number 8, and this is the structure of formyl glycinamide ribosyl 5-phosphate, which is formed in the fourth step. Then the product which is formed in the fifth step is formyl glycinamidine ribosyl 5-phosphate. So here glycinamide becomes glycinamidine and here the donor is glutamine. So there is formation of formyl glycinamidine ribosyl 5-phosphate. This is the fifth step, which also involves ATP and responsible for contribution of nitrogen present at number 3. In the sixth step, ATP is involved. There is just ring closure. Imidazole ring is formed. So there is formation of amino imidazole ribosyl 5-phosphate. So there is ring closure. In the seventh step, the product form is amino imidazole carboxylate ribosyl 5-phosphate. So carboxylate means carbon dioxide is involved here. So it is responsible for contribution of carbon present at number 6. So carbon dioxide comes here and carbon number 6 is contributed. In the step number 8, there is formation of amino imidazole succinyl carboxamide ribosyl 5-phosphate. Here the donor is aspartic acid. ATP is also required and this aspartic acid contributes nitrogen 1. So here there is formation of amino imidazole succinyl carboxamide ribosyl 5-phosphate. In the ninth step, the fumarate is removed to form amino imidazole carboxamide ribosyl 5-phosphate. So this is the structure of amino imidazole carboxamide ribosyl 5-phosphate. In the 10th step, there is formation of formed amino imidazole carboxamide ribosyl 5-phosphate. So here the donor is formyl tetrahydrofolic acid, which contributes carbon number 2. And this is how carbon number 2 is donated by tetrahydrofolic acid. And in the 11th step, there is ring closure and there is formation of adenosine monophosphate. So these are the various 11 steps of purine biosynthesis, and further from this adenosine monophosphate, AMP and GMP are synthesized. Students, please note that the structures of the various products that are formed are not important. You have to just remember the donors and the name of products. So I hope this video on purine biosynthesis will help you to understand this purine biosynthesis pathway. And if you find this video useful, please like this video, share the video, and thank you for watching and happy learning.